Now, looking at PFK1 and ATP, what we see is that at low levels of ATP, our curve of activity versus the substrate concentration, fructose 6-phosphate, is kind of getting close to Michaelis-Menten kinetics. There is a little bit of sigmoidal activity that we see here. However, when we go to higher levels of ATP, we definitely see some sigmoidal character to it. It pretty much loses all of its more Michaelis-Menten type hyperbolic kinetics. Now, the reason for this is that there are two binding sites for ATP on the enzyme. We have a substrate site, which is a relatively high affinity site. That means that it doesn't take a whole lot of ATP to bind to the substrate site. We have a low affinity regulatory site, meaning that it takes a relatively high concentration of ATP to bind to the regulatory site. That's why we see with our high ATP levels, we see this distinct shift, getting a sigmoidal character to this curve. Now, basically as ATP levels rise, we'll see binding to regulatory site and inhibition of PFK1. When ATP drops, well, we want glycolysis to be active. So ATP levels start to drop and ATP dissociates from the regulatory site, and as a result, it simply serves as a substrate for the enzyme. One of the questions that often arises is, where does the AMP come from? Because as we see in glycolysis, we have ATP going to ADP. When muscles use ATP, when actin and myosin use ATP as a source of energy, the ATP is converted to ADP. So where is the AMP actually coming from? And the thing is that two molecules of ADP can be combined together to make a molecule of ATP and AMP. That ATP is then going to be used pretty quickly, particularly in muscle where it's going to be pretty rapidly used as a source of energy. And thus, we see a fairly rapid increase in AMP. ADP levels really don't change that much as a result of this reaction, whereas AMP can rise pretty quickly as a result of this. So this is how we get AMP to serve as an activator for PFK1, basically making it really quickly from the ADP that is being produced as a result of the hydrolysis of ATP. Fructose 2,6-bisphosphate is a very important regulator of both PFK1 and fructose 1,6-bisphosphatase. And we only see this in the liver. And basically it is produced from fructose 6-phosphate. We take a small amount of this fructose 6-phosphate out of glycolysis to make fructose 2,6-bisphosphate. And what that does is it is a potent activator of PFK1 and thus activates glycolysis. It's an inhibitor of fructose 1,6-bisphosphatase and thus an inhibitor of gluconeogenesis. So if we look at the general role this plays, here's our fructose 2,6-bisphosphate, activation of PFK1, inhibition of fructose 1,6-bisphosphatase. Therefore, it's going to activate glycolysis and it's going to inhibit gluconeogenesis. So let's take a look at this in a little bit more detail. How this works is that here's the activity of PFK1. In the presence of fructose 2,6-bisphosphate, the activity, if we kind of continue this down here, looks pretty much like a hyperbolic type enzyme. Whereas in the absence of fructose 2,6-bisphosphate, we see that it requires a fairly high concentration, well over a millimolar concentration of fructose 6-phosphate to get any sort of activity out of this enzyme. Basically, even at 1 millimolar fructose 6-phosphate, we have an activity of PFK1 that's about 10% of Vmax. The thing to notice is that in the presence of fructose 2,6-bisphosphate, the concentrations of fructose 6-phosphate that are needed to achieve 50% of Vmax basically are less than 0.1 millimolar. So we're talking a fairly low concentration of fructose 6-phosphate needed as long as fructose 2,6-bisphosphate is present. Let's look at the activity of the fructose 1,6-bisphosphatase. So in the presence of fructose 2,6-bisphosphate, this is when we expect glycolysis to be active, we need pretty high concentrations of fructose 1,6-bisphosphate in order to achieve any sort of activity. Basically, we're up here at approaching 100 micromolar. And this is a relatively high concentration for fructose 1,6-bisphosphate because it tends to be used fairly actively by other enzymes in the glycolytic or gluconeogenic pathway. In the absence of fructose 2,6-bisphosphate, what we see is that the concentration needed to achieve 50% activity is extremely low, in the low micromolar range. So in the absence of fructose 2,6-bisphosphate, we see a lot of activity for fructose 1,6-bisphosphatase. So where does fructose 2,6-bisphosphate come from? Well, it comes from fructose 6-phosphate, and there is an enzyme that is able to both produce it and break it down. It's actually a bifunctional enzyme. It's a kinase, PFK2, and a phosphatase, fructose 2,6-bisphosphatase. And as we can see from this general scheme here, if we have PFK2 active, we're going to be making fructose 2,6-bisphosphate. If we have fructose 2,6-bisphosphatase active, we're going to be breaking it down. And the control over this enzyme depends upon phosphorylation. So in the presence of glucagon basically a hormone that indicates low blood glucose, we are going to phosphorylate this enzyme, and we get this form down here. When it's phosphorylated, the phosphatase form is active, meaning that it's going to be breaking down fructose 2,6-bisphosphate. That's going to take away the activator for PFK1, the inhibitor for fructose 1,6-bisphosphatase. Thus, we're going to see gluconeogenesis activated. If, on the other hand, we remove that phosphate, we have PFK2 active. Therefore, we're going to be making fructose 2,6-bisphosphate. That's going to activate PFK1 and glycolysis, inhibit fructose 1,6-bisphosphatase, and thus inhibit gluconeogenesis. Now, the kinase is, we basically convert this into the kinase form in the presence of insulin. So insulin is the activator here, whereas glucagon is going to activate the conversion to the phosphatase form. So ultimately what we see here is regulation by hormones. Insulin is activating glycolysis. Glucagon is activating gluconeogenesis. And a lot of that is being achieved by regulation of this one specific enzyme, this bifunctional PFK2/fructose 2,6-bisphosphatase enzyme. And whether or not it's phosphorylated will tell us which of the activities this particular protein is going to have. Pyruvate kinase is regulated, and it kind of represents a little bit of an anomaly in terms of regulation because it sits at the end of a pathway. Normally, regulation occurs at the beginning of the pathway, which we saw with the small amount of regulation over hexokinase, and then the large amount of regulation over the commitment step of the pathway, PFK1. The reason that we regulate pyruvate kinase is that it is technically part of a substrate cycle. If we have, for example, PEP going to pyruvate, and that's pyruvate kinase, well, pyruvate goes back to oxaloacetate and then to PEP using the pyruvate carboxylase and PEP carboxykinase enzymes. So if we are doing this portion, we need to have pyruvate kinase inhibited. So that is the reason for the regulation at pyruvate kinase. Now, pyruvate kinase has some interesting allosteric activation. It's activated by fructose 1,6-bisphosphate. This is what we call a feed-forward activation because fructose 1,6-bisphosphate was produced way back at PFK1 about 6, 7 steps ago in the glycolytic process. And this is analogous in my mind to thinking of glycolysis as a typical river in the western United States that tends to get dammed, and you have basically you convert a river into a series of lakes. And what we have is we've got this dam here this is PFK1, and then we have this lake sitting below it, and then we have another dam at pyruvate kinase. And if we want water to be moving along this pathway, basically moving in this direction, well, the water that's here essentially has to come forward and activate pyruvate kinase, basically making it spill water through that dam in order to get water moving in this direction. So I know this is a pretty odd analogy for those of you who may not realize the plumbing of the western United States, but this basically explains why we have this activation of pyruvate kinase. We want to if levels of fructose 1,6-bisphosphate start to rise, well, that means that we need to activate pyruvate kinase in order to use the phosphoenolpyruvate that's sitting right there and cause everything else by equilibrium to move towards the phosphoenolpyruvate. There's also some allosteric inhibition, ATP. If ATP levels are high, we inhibit pyruvate kinase. Acetyl-CoA also inhibits it, and we'll see acetyl-CoA when we get to talking about the citric acid cycle, but ultimately what happens is that pyruvate is converted to acetyl-CoA. If acetyl-CoA levels are high, then it means that we need to stop converting phosphoenolpyruvate into pyruvate. There are a few other regulatory points as well. We see that alanine, because alanine can be converted by transamination into pyruvate, it serves as an inhibitor. Long-chain fatty acids also have a regulatory effect. I'm not going to go into that right now, but we will see that when we get to the control over fatty acid synthesis later. If we look at what goes on specifically in the liver, glucagon happens to be a regulator of pyruvate kinase, and it does so by phosphorylation. So we see that in the presence of glucagon, we have this enzyme protein kinase A that sticks a phosphate on pyruvate kinase, puts it into the inactive form. A protein phosphatase will remove that phosphate, converting it back to the active form that we see here. The liver has all this allosteric regulation as well, but it has this one other impact as well, and that is glucagon. And glucagon has pretty much its sole impact on the liver. It does impact adipose tissue as well, but there's a lot of things that specifically occur in the liver in response to glucagon. So we say that glucagon is largely a hormone that has a tremendous impact upon the metabolism that goes on in the liver.